Hi, everybody. Welcome back to Industry Corner Live at IEEE Pulse. My name is Chad Andresen, the editor-in-chief, and we have a wonderful guest today. We have Vijit Sabnis, CEO and co-founder of Adaptyx. Vijit, how are you?
I'm doing well. It's great to be here. Thank you.
Yeah, well, we've had wonderful connection and us bringing in together to talk about where Adaptyx is going. But I always want to start a little bit about your background and how you got to be CEO and co-founder.
A little bit about myself. So I've got a PhD from electrical engineering at Stanford University, and I've been connected with Stanford ever since graduating from there more than 20 years ago. My background is in optics and optoelectronics. I started off at another startup here in the Palo Alto area doing telecom. I then moved over to starting my own solar energy company called Solar Junction, which was a spin-off from Stanford. This is my former PhD advisor's research that we commercialized successfully. And that was a great venture-backed company that was acquired back in 2014. I then had a wonderful experience being a venture capital investor for about 5 years. And that was a major transition in my career where I was able to work on everything that I love about technology. The firm I worked with was very broad and deep across all areas of technology. And one of the areas that I had an opportunity to work in was in health technology. And the firm I worked in was very forward thinking about the use of AI and data in healthcare. And one of the things that I really took on as one of my passions was, could we find new technologies that would generate data that we could use, across healthcare in ways that were not always obvious. This is about a decade ago, and back then we didn't call it AI, we called it big data, and we can all chuckle about that. But the whole idea was to generate data with the conviction that algorithms which obviously have progressed greatly over the last decade, but those algorithms could teach us a lot about our bodies. And so we were pretty early in thinking about data fusion. And today we think about that in terms of like multimodal AI and how we can then use those techniques to really shed insights, that are unusual or things that wouldn't be easily predicted. And one of the things I became very passionate about was the need for collecting more data about our bodies, about our health, in ways that were really much more expansive and much more convenient than, for example, conventional blood testing. And I became really, really passionate about, for example, about the continuous glucose monitor, which you and many of your viewers may know that this is a small patch that is attached to the arm, and it has a probe that goes underneath the skin, which can measure the molecule glucose continuously. And this is extremely valuable for diabetics. And at the time when I was an investor, I began to look for technologies that could measure not just glucose, but really any molecule that was relevant to our health. And that ultimately became the foundation for, the technology that we're developing at Adaptyx.
Oh, okay. So we've got the beginnings of from founder or co-founder in an early startup, investor, and then back into the startup realm.
That's right. Yeah.
That's a great combination. I think that's ideal. Plus PhD on top of that. So your amount of wealth of knowledge is going to be great here as we go. What is Dactyx doing? What is coming out of all of that experience, and what was the original inspiration in the labs to get the unlock this new ideas
Absolutely.
that you're bringing forth?
Yeah, so my fundamental thesis is that we need to collect a lot more data about our health, and we're seeing tremendous demand from consumers these days of wanting that data. So, for example, we have the Aura Ring, the Whoop wristband, the Apple Watch, the Fitbit, and these are developing and delivering to us really amazing insights about our health. heart rate, HRV, steps, recovery, and that's been absolutely wonderful. But when it comes to biochemical data, we're really limited, for example, to blood testing. And our vision at Adaptyx, and one of my driving really visions here was I want to bridge our clinical care that we get from our physicians, and that's typically done when we're sick or maybe done at an annual checkup. But there is a gap between our everyday lives and how each of us manages our health. There's a gap between that clinical care and us managing our everyday health. And that's where like really my inspiration came in to co-found Adaptyx. I want to build a device. This is going to be a patch that goes on the arm or the abdomen that collects very specific data that can help me or any of us as users manage our everyday lives. But also, that data can be shared with our physicians to really help our physicians understand what's going on in a day by day basis with this particular patient. And how do we take those clinical needs and that clinical care and really help translate that into maybe a drug someone's taking, maybe a lifestyle adjustment or intervention that someone's taking. And so this continues today to be, I think, a large unmet need. To me, the big limitation is ultimately technology. And so I was looking at this from from an investor standpoint, starting about a decade ago. I became enamored with the continuous glucose monitor. I think it is absolutely a game changer in folks being able to understand and ultimately manage their metabolic health. And I'm really delighted over the last 10 years, and particularly the last 5 years, of that market and that understanding really growing tremendously.
Mm.
And so I, as an investor, was looking for a platform technology that could measure not just one other molecule, but really, frankly, any molecule that we needed to measure continuously in real time. And so there's a couple of really interesting things here. Our bodies, as we know, they're dynamic. They're changing all the time. Each of us feels differently on a day-by-day basis. How I feel today may be a little bit different than tomorrow. Our bodies are dynamic systems. And so while blood testing is incredibly valuable in understanding a particular snapshot in time of what's going on in our body, we really have no technologies that are allowing us to understand how things are changing over time. Think, for example, about hormones. Our hormones are shifting. These are
Mm.
really, really relevant for all sorts of different conditions, whether you have a thyroid condition, whether you're under periods of stress. For example, one of the things we're able to measure at Adaptyx is the stress hormone cortisol. There's obviously sex hormones like testosterone, estrogen, progesterone, which are changing over the course of a day, over the course of a month. These are things that are dynamic, that are affecting our health in really, really big ways.
Mm.
And our healthcare system today, through blood testing, is really unable to capture those changes. Another really good example are drugs. Each of us metabolizes drugs differently, and oftentimes dosing them and really being able to change the titration of these drugs over time. That's another really unmet need, in the healthcare system today. So I was looking for technology that could tackle this. And ironically, when I decided I really wanted to go out and build another company, I got reconnected with a gentleman named Tom. So Tom is a professor at Stanford. He's been there for more than a decade. And as it turns out, Tom and I were fellow graduate students in the electrical engineering program at Stanford a long, long time ago. And so we were acquaintances there. Tom is a little older than me, and so we overlapped a bit in grad school. And so, a couple of decades later, we got reconnected through a mutual connection who said, "Hey, I think you guys are interested in the same things. And you guys ought to talk, and I think you'll enjoy getting reconnected with each other." So this was way back in the middle of 2019, and Tom began to tell me about his research that he has been developing at Stanford, and prior to that at UC Santa Barbara. And Tom has developed a new technology, which we're calling today at Adaptyx programmable molecular switches.
Hmm.
And these are synthetic strands of DNA that can be integrated into a patch-like device. And what's really exciting about these programmable molecular switches is we can design and develop these switches to measure different analytes in our body continuously in real time. This is really different from today's glucose sensors. Glucose sensors today rely on an enzyme called glucose oxidase, which translate glucose into a measurable electrical signal, which is how continuous glucose monitors operate today. Tom's developed a platform with these programmable molecular switches, which are based on these DNA molecules called aptamers. And so, what we're doing at Adaptyx is we've developed this into an entire chemistry platform using lab automation and now increasingly machine intelligence, which allows us to design these programmable molecular switches to be able to measure small molecules, hormones, proteins, electrolytes. This list goes on, and we can now measure these different classes of molecules continuously. And so ultimately, what we're trying to do at Adaptyx is taking conventional blood tests, but move them into a small patch that's going to go on the arm or the abdomen and measure multiple molecules continuously in real time. And our goal at Adaptyx is to bring these sets of molecules into applying this to clinical applications and over time to helping consumers manage their everyday health.
That's incredible. It sounds like it's a laundry list of things to monitor and measure. What is the single physiological blind spot in medicine today that Adaptyx is in a unique spot, specifically to eliminate? Maybe that's CGM or beyond, but what is that one, the big one? What's the biggest blind spot?
Well, look, there's a lot of different things that we can do here, and we want to attack all major categories of health with our technology over time. But we spent a long time thinking about where can we have the largest impact, and it really comes down to, in the next several years, two major areas. The first one is in continuous hormone monitoring. Hormones, as I mentioned earlier, are dynamic. They're changing over time. And one of the areas that we're going to focus on first is in the continuous measurement of the stress hormone cortisol. Cortisol, as you know, is a stress hormone. Actually, there's been a lot of media attention in it over the last couple of years. Cortisol is a master regulatory hormone. Every single organ system in your body has a cortisol receptor.
Hmm.
And this is something that's a little bit underappreciated, but cortisol does so many things in our body that affect our everyday health, as well as some of the major chronic conditions. And I'll give you just a little bit of a glimpse of why we're so excited about this. Cortisol is a major regulator of metabolism. It works alongside insulin to control our glucose levels in our body. So, insulin, as we know, helps glucose go directly into cells. And cortisol, which we call a stress hormone, in times of stress, it actually acts against insulin to increase our glucose levels. So, for example, when you need to run away from something, or when you need to concentrate very heavily, or when you're, for example, in an argument with someone, cortisol over time is going to turn on and it's going to increase your glucose levels. One of the things that's really underappreciated is in chronic conditions like diabetes, as we're learning more about hypertension and obesity, chronically elevated levels of cortisol can be an aggravator of those conditions. And so we're developing our cortisol sensor to help manage and understand those conditions. There's also, for example, some rare diseases called Addison's disease and Cushing's disease, where cortisol is either produced way too high or actually not produced at all. And these are actually major health conditions for these folks that need to be addressed. Cortisol, again, is a marker that is changing throughout the day. And today's blood testing or saliva testing only allows us to measure a snapshot in time. And so we're with our device able to measure cortisol rhythms throughout the day and provide that interpretation back to physicians, which we think are going to help us understand and treat these rare conditions, as well as some of the major chronic diseases like diabetes, hypertension, and then even in the future, obesity, mental health, osteoporosis, a number of autoimmune conditions. So again, we're really excited about starting off with continuous hormone monitoring with cortisol. And then over time, we're going to be adding to our device testosterone and even in the future, progesterone and as well as estrogen.
Mm.
Another major application we're developing today at Adaptyx is in the management of heart failure patients. Heart failure affects about 7 to 8 million Americans, about 65 million people globally. It's a chronic condition in which there is no treatment. And there's no cure to that disease. So it is something that our healthcare system is really actively involved in treating these folks as they progress through their journey. And one of the really exciting things that we're doing um, is developing the ability to measure kidney markers like creatinine, urea, some of the key electrolytes like potassium and sodium, as well as a heart stress hormone called NT-proBNP. So we're developing that panel, which is going to be able to measure over time all of these markers continuously. And we're going to help physicians use this information to titrate medications that are given to heart failure patients. This can allow physicians to allow these patients to get to the maximum levels of doses of these medications to keep them safe, to keep them out of the hospital, to also understand how their heart failure may be progressing over time. And by measuring this set of biomarkers, we can actually predict when a patient is going to have what's called a decompensation, which is when their heart failure gets to a very quickly to a position where they need to go to the hospital in an emergency situation. We can actually foresee this a couple of weeks ahead of time
Mm.
by doing this real-time tracking and help these patients stay safe, get to the physicians when they need to. And this is an example where that continuous sensing, monitoring these trends over time is a real big win compared to what once in a while reactive blood testing is able to offer.
No, it sounds like sounds like it's a big blind spot. The hormones, and like you said, the heart failure, stress, uh, biomarkers that you're able to see. I think there's a big spot where pharma comes in. How do you see your guys selves, this kind of technology amplifying new drugs or amplifying new studies, or even old drugs, and helping really establish where they're the
Yeah.
best at?
Yeah. There's a few areas that we would love to collaborate with pharmaceutical companies. The first is in clinical trials. So when we collectively run clinical trials, pharma companies have to go out to their patients, run blood tests on them, bring that data back, and they're looking at that in a very coarse way. With our device, for example, if a pharma company wants to understand how is someone's kidney function changing over time based on a new drug that's being tested, our device can collect that data, for example, continuously. So, for example, we've been having discussions with different companies about the value, for example, of measuring electrolytes or creatinine or urea or cystatin C. And we're going to be able to give these pharma companies much, much more granular data than they've been able to collect with once in a while blood testing. So that's one particular case where providing very granular, continuous data in clinical trials is an area that I think can be useful for pharmaceutical companies. A second one is in the fundamental development, for example, in small molecule drugs. Small molecule drugs, that may be developed sort of in that discovery and preclinical phase. We can develop our sensors to measure the concentration levels of those small molecule drugs, and we can help pharmaceutical companies understand what they call PK and PD, so pharmacokinetics and pharmacodynamics. Instead of using once in a while blood testing to understand drug metabolism or the body's response, biomarker response to a new drug, we can actually measure the concentration of that drug in real time, or maybe some of the body's response, for example, kidney markers or liver markers in real time in response to that drug. So the development of really granular PK and PD models for pharmaceutical companies is something that our sensor can enable these companies to do. And this is something that's never been available to these companies, and we're excited to speak to companies about partnering with them.
I think that's a big deal that everyone could, anybody could be a part of a clinical study and even feel a little safer being able to monitor those things as well.
Chris, and sorry, Chad, one of the things that we're able to do is with our chemistry platform, we can develop new sensors very, very rapidly. This is something that we've invested a lot of time and effort in building and designing and developing these programmable molecular switches. In about 1 quarter, we can take a new molecule that we look at, understand it, go develop it with our models, and go develop a prototype sensor in about 3 months. And this is something that's really unprecedented, that in the world of continuous sensors has not been possible before.
That's incredible. So there's almost no limit with the things that can be thought of or as drugs evolve, what you want to monitor and measure. Well, why don't we talk a little bit about how the technology a patient might use it in the future?
Yeah, so we're developing, again, this is a patch-like device. Ultimately, it's going to have a form factor that is similar to a continuous glucose monitor. So I've got my Apple AirPods case here, so it's going to be around that size, and it will go around on your arm or on your abdomen. It'll be self-applied at home, and it will have a probe that is inserted very rapidly just underneath the skin with an applicator. So an applicator is a device that injects that with a guide needle just underneath the surface of the skin, a few millimeters deep. You don't really feel anything after that probe has been inserted underneath the skin. And the patient or the consumer will wear this, depending on the application, over the course of a few days. For example, if we want to understand someone's cortisol rhythms, wearing the device for 2 to 3 to 4 days gives us plenty of time to really understand someone's cortisol rhythms. If it's, for example, a heart failure patient, they may want to wear the device for a couple of weeks or maybe in couple of week batches over the period of several months to really help them understand, their medications, how we want to adjust their medication over time, how, for example, NT-proBNP, the heart stress hormone, is changing depending if they've had a change in their condition. So it's going to be similar to how people use a continuous glucose monitor. But in some cases, it may be just for a period of a couple of days. And in other cases, it may be ongoing use, similar to how diabetics are using the CGM.
Very interesting. So the technology is short term, right? It's meant to be disposable, replaceable,
That's right.
and it's continuous. I guess when you talk about going to the FDA and seeking regulatory clearances, how does that pathway look for you guys? Are you trying to get a single indication at a time? And then what kind of clinical data do you guys have to use to support that as well?
Yeah, so we believe our device will be a class 2 device, either a 510(k) or de novo approval. We're still in discussions with the FDA on exactly which pathway that's going to go down. But this is generally considered a very safe device. Again, it's minimally invasive. It's relatively pain-free, particularly after that first probe insertion. What we're going to do with our device is kind of go one set of molecules at a time through the FDA. We're going to first start, again, with the cortisol monitoring as well as the heart failure monitoring applications. Going through the FDA, is a process, as it is for all medical device companies. We'll be kind of collecting clinical data with these markers on our devices. We're going to be showing that our the value that we're measuring on our device matches, for example, blood testing. So, for example, the way our clinical studies are going to be laid out is we're going to be collecting our data with our device on a cohort of patients. And those patients will be wearing our device over a period of time, and we'll be making blood measurements periodically on these patients. And what we'll be doing is refining our models for the data coming off of our device and developing models that enable us to calibrate the data from our device so that it matches the blood values. And what's exciting about the way our sensor works is that our probe, which is just located underneath the surface of the skin, measures analytes in dermal interstitial fluid. So interstitial fluid is the fluid in between cells, and in this case, we're probing the dermis. And the capillaries, which carry blood, as you know, they transport molecules in blood through the capillary walls into the dermis relatively easily, particularly for smaller molecules and smaller proteins. And so the correlation between analytes in dermal interstitial fluid and blood is very, very good. It's very robust. In our clinical studies, which we have started here over the last year, we'll be proving that the analytes that we measure with our sensor in ISF match well with blood. And so this is taking a similar clinical playbook that the folks who have developed the continuous glucose monitors have done over the last 15 years, 15 to 20 years. We're going to be doing clinical studies that are relatively similar to what those folks have done. And our plan commercially is to go from one set of molecules to another set of molecules over time. Again, as I mentioned, we're focused on continuous hormone monitoring as well as heart failure monitoring as kind of our major foci in the coming years ahead.
Those are good groundbreaking steps to make. When you looked at, and you have been looking at many types of analytes to observe, is there one that's the hardest to measure continuously? And is that something on the back burner that you guys have as well?
Yeah, well, my dream and vision is I want to be able to measure very low concentration hormones.
Mm.
Things like insulin, estrogen, and progesterone are down in that picomolar range. And that is a challenge. It's on our roadmap. This is something that we're actively working on. I think if we're able to do that, or if the world is able to do that, it is massively, massively valuable. Those are even placeholder molecules. If we can measure those types of molecules, there are many, many other hormones and other very low concentration analytes that are incredibly valuable. I would love to be able to do insulin, progesterone, and estrogen. Those are markers that we're working on. They are within reach.
Mhm.
They are not something that we think we're going to be able to commercialize in our first set of products. But those are something that are very near and dear to my heart that I would love to be able to go off and measure continuously. And there's a number of technical reasons why that's challenging. But those are sort of holy grail molecules that I think if we could measure the impact on clinical and consumer health, as well as just our fundamental understanding of physiology, are going to be a really big breakthrough.
Yeah, I think that's unlocking a whole new view into the body. I think that you could definitely think of new drugs, like I said, and new even neuromodulatory devices that can do bioelectronics.
Absolutely. Yeah.
Yeah, be effective in there. That's a world I live in, so I'm thinking as we speak. But with continuous glucose monitoring, I don't know if there was that big of a gap for physicians to trust it and not trust it.
Yeah.
But do you think that there's going to be a gap of trust, as physicians begin to trust your technology and their patients? And what do you plan as your vision to help, increase that as well?
Yeah, look, I think this is fundamental to the deployment of any new technology, and developing trust with physicians at Adaptyx is our absolute number one an initial goal. If we can't get physicians to trust our numbers and the interpretation we're providing, then we're not going to be able to ultimately help the patients that we dearly want to help. We're going to be doing a lot of clinical studies. We're going to be building a base of literature, based on clinical studies that key opinion leaders, endorse and want to see. Our strategy as a company, when we look at the range of analytes that we could deliver with our technology, one of the things that we want to do is really focus on molecules where blood testing or the timeliness of the data, is not possible today. So, for example, with hormones, for example, cortisol is changing throughout the day. Even though there are very well-developed conventional testing with blood or with saliva or with urine for measuring cortisol, those tests are fundamentally challenged in providing the interpretation that physicians need because they're measuring just a single point in time, or in the case of urine testing, kind of an integrated look at cortisol over the course of a day. We're going to be measuring cortisol. We're going to be delivering a curve throughout the day, and that diurnal variation is what your body is producing. And so when we look at, physicians looking at our data and ultimately trusting our data, we are providing the ground truth in what cortisol is doing in the body. So even though we are a departure from conventional testing, I would argue that conventional testing is giving us a very, very incomplete and frankly inconclusive view of cortisol. So I think we're actually going to have a little bit of an easier time in getting physicians to trust our data because we are delivering ground truth and that true diurnal change. Now we're going to have to provide the data to physicians, and we're going to be doing that by providing these rigorous clinical studies. But that's been one of our strategies in finding where are we as a company going to have maybe a little bit easier time developing trust with physicians, and we're going to be doing that by delivering something the data that they don't have today, which ultimately is what your body is providing through that cortisol, continuous curve.
No, incredible. I think my next question comes to, you talked about molecular switches. Is there an active biological component in the device? And is it, maybe a combination device? Or is it something more based on even your past with optoelectronics, rather than maybe
Yeah, so
active biologics?
Yeah, so these are passive DNA structures that form these switches, and what Professor Tom So developed at Stanford, and really, I think his pioneering contribution to the entire field is, one thing I should point out is that when we get a blood measurement done today, the way a molecular clinical assay works, and this is a very general statement, but there is a binder that basically you flow blood over this assay. And this binder is specific to a particular molecule. And that binder captures this molecule. It never lets go, or it lets go after a very, very long time. But let's say the first order, it never lets go. And that then allows a measurement to be made. That could be an electrochemical measurement. It could be a fluorescent measurement. It could be a colorimetric measurement, for example. And that's how, for example, again, this is oversimplification, but that's in a way how blood testing works. We capture a molecule, and through some means, we measure this. If we want to make a continuous measurement, you have to think about a completely different paradigm. In continuous glucose monitor, there's an enzymatic reaction which is being recycled. That's how CGMs work. But unfortunately, enzymatic sensors are really, in the terms of continuous sensing, they're really limited to just a handful of few molecules: glucose, lactate, ketones, alcohol. But that is not a generalizable, continuous molecular solution for sensing. What Professor So pioneered, starting initially at UC Santa Barbara and at Stanford, is the ability to develop these DNA-based programmable switches where you've got this DNA switch where it binds to a molecule, a target molecule of interest. It binds to that for some period of time, and then it lets go. And then another molecule comes by, the target molecule, it binds again. So these are what we call reversible molecular switches. So they
Mhm.
bind and they let go. They bind and they let go. And we design these switches so that binding and unbinding happen at about approximately the same rate that these molecules are changing their concentration in the body. So we're able to tune these switches so they bind over the concentration range present in the body. They also bind and unbind at approximately the same rate as we expect to see these concentrations changing in the body. So it's the what we call the binding affinity as well as the binding kinetics that we're able to engineer with these molecular switches. And that's the real pioneering breakthrough that Tom, as well as as other workers in the field, have really been developing over the last, 15 to 20 years. And this is a platform. This is not limited to just one molecule, but we can design the structure of these molecules to work with small molecules, to electrolytes, to peptides, to proteins. And so we really think, we have a large canvas of molecules over the next 5 or 10 years to develop these sensors to work across. And we have purpose-built our sensors to measure not just one, but multiple molecules, simultaneously and continuously. In fact, our prototype sensors in our lab can measure up to 16 molecules continuously.
Wow.
Commercially, we're gonna start off with a smaller set of molecules, but today we're able to do up to 16 molecules, and in the future, in principle, we could measure a lot more. Um,
Wow.
the other thing that's really exciting, Chad, and this is oftentimes, I think underappreciated. Measuring one molecule is really, really valuable. For example, glucose. But the really valuable thing happens when you start to measure multiple molecules. So, for example, if I measure, let's say, 4 molecules continuously, I can look at how I can look at the relationship of one molecule in conjunction with others. And you can start to look at all these different permutations. For example, you know, how does cortisol interact with glucose or lactate or electrolytes? And you can start to think about in a very patient relevant way what is the relationship between these different molecules? That's where
Mm.
AI, for example, comes in. You can take the data off of our sensor and you can start to look at blood records or other, for example, health data that we're gathering. For example, heart rate variability. We're really excited about looking at cortisol data in conjunction with heart rate variability and what that means for athletes, what it means, for example, in sleep monitoring. How does it affect different aspects of our physiology? So this is not an exaggeration. As you look at more molecules continuously, and as you combine that data with other types of data that you're collecting from other wearables or other medical records, the value that we're going to get is exponentially more interesting and more valuable. And these are things that we cannot really predict and ascertain today.
Mm.
But I think there's going to be an explosion in value coming out of multimodal, like health data fusion. And we want to be an important part of that overall ecosystem with our continuous data from our sensors.
No, it sounds like you're on that path. What does the next few years look like? When do what kind of development, how long are your clinical studies and kind of expectation to market are you guys looking for?
Yeah, so over the next 2 to 3 years at Adaptyx, we're focused on a lot of clinical studies, collecting data on our device, making our sensor is robust. This is quite a challenge. Our goal is to build a factory calibrated sensor where we can develop our devices, ultimately send them out to a patient, and they're going to work directly out of the box. So we're going to be collecting a lot of clinical evidence, making sure that our manufacturing, our calibration of our devices is, as robust as it needs to be able to serve these patient populations. Secondly, in parallel with that, we're going to be focusing on different pathways of miniaturizing our device. Today, we're a lean startup with just about 15 to 16 people in our company. Our device is a little bit larger at the prototype level. We're going to be focusing on miniaturizing this down, and over time, we'll ultimately get to that small CGM-like form factor, but it will take us some time and obviously a lot more capital to get there. But we've got a really exciting path, to really get there over a large number of molecules. And right now, our focus at the company is, let's get that clinical evidence. Let's make sure it's robust. We're in the process of developing, a group of key opinion leaders who are excited about guiding us and, just really making sure that our data is robust.
Oh, that's great. Well, we're at the end, but I always have a question for every guest. And I think with your expertise background, seeing you've touched a lot of the things, what is the some advice that you would give to a young entrepreneur or a PhD student trying to start a company? What's something you wish somebody had told you? What have you learned in your experience?
Yeah. I think that a lot of times, particularly maybe folks who are going through their PhDs and whatnot, um, oftentimes people are really reticent to go off and pursue some really exciting ideas. Maybe that's at a really exciting startup, or maybe they've got an idea that they want to pursue. Um, there's never a good time where you feel ready. There's never a good time where you're going to feel like the risk is really manageable. And I feel like I really would have benefited from just go for it. The risk is never as much as as you oftentimes perceive it to be. And folks oftentimes, and myself included, oftentimes overanalyze these things. You don't know until you jump into things.
Mm.
And so being agile, really going for things, you're going to learn more by doing, rather than sort of observing. And so, I really, I was lucky that I got some good mentorship while I was in graduate school at Stanford. And Stanford's a pretty special place in that there's a lot of people thinking about entrepreneurship and really taking some of the most innovative technology and finding ways to go off and commercializing that. And I felt really lucky being at Stanford in the late 90s and in early 2000s, which I think were a really big transformation here in Silicon Valley, as we all know, with the internet and things only growing
Yeah.
exponentially since then. But I think just students and young people all around the world, really need to take heed of that. There's talent everywhere, and I was in India, visiting family just over the holidays, and there's just so much talent there, and there's so many folks really anywhere who can contribute positively, to society. And in my career as an electrical engineer, I didn't start out in health. I didn't start out in medical devices, and I've been very fortunate, here in Silicon Valley and with, I think just a really good group of friends and mentors to, really have the opportunity to focus on really, really big, important problems and finding a way to, just go for it. To get a group of people together and really tackle big, almost impossible problems, and to find your path. And I've done that in telecommunications, working at a company that was really early in the development of the field of silicon photonics. Silicon photonics is a really big, very big, impactful industry today, affecting, data centers. I was really excited to be part of that early field of a development way back in that 2003 to 2008 timeframe. I was really lucky to develop a new solar cell back at Solar Junction starting in 2007, which led a wave of, I think, solar being really ubiquitous today in the world. And then I was able to have an opportunity to pick another big problem in healthcare, which is, in my opinion, data generation and the utility of that data, empowered by AI, which is what I'm working on today. And so, my advice to young people is always be learning, take risk, go off and tackle some of the biggest problems in society. Um, and don't be afraid to move out of your field.
Mm.
I've been in a position where, every 5 or 7 or 8 years, I'm sort of switching to where the problems take me. And so problems and problems that need to be solved have guided my career rather than my educational background. And don't be afraid to go off and do that. You'll find that technology moves fast. What you know today will, may be less relevant 2 to 3 years. It's somehow that time window, shrinks.
Mhm.
As my career has progressed, you always have to be agile, and you always have to go towards the problems and not necessarily what you're comfortable with or what your education has maybe grounded you in.
No, it's great feedback. I always find myself wanting to move out of my space of active implantables, but it always just keeps
Yeah.
calling me back as well. But I'll get to that spot one day, and I love that advice and it coming from such an accomplished individual as yourself. And I can't wait to see Adaptyx explode and get used to being the norm in this life. I think that's where we're heading.
Well, thank you so much. I appreciate that encouragement and the time to speak with you and your audience today.
Yeah, thank you, Vijit. Have a good day.
Okay, thank you.